There are 20 total amino acids in this world to make all of the different proteins in this world. That's kind of a small alphabet and a lot of words. A protein is two or more amino acids bonded together. They are bonded by a covalent bond, but this bond gets a little bit more of a specific name. We call it a peptide bond. A polypeptide is a string of amino acids bonded by peptide bonds. This is not officially a protein, but it's the start of a protein. These amino acids are bonded together in dehydration reactions in which water was removed to build or synthesize a larger molecule. This could also be called anabolism, the building of a larger molecule. I think of protein structure like those folded paper fortune tellers we made in second grade. What's your favorite color? What's your favorite number? Right? And then you open up the fortune teller and it tells you who you're going to marry. I married Mr. Sherman. Despite your potential spouse, the fortune teller, it's an important analogy since it's folded specifically to create the final product, right? If you don't fold it right, it's not going to work right. In fact, there are different levels of folding to create a final product like a protein. And proteins are kind of similar in that they're folded 3 times to create a final complex and a very specific structure. These 4 levels of folding are known as the primary, secondary, tertiary, and quaternary levels of folding. And each time a protein is folded, there are hydrogen bonds holding the folds in place. We referred to hydrogen bonds in a previous mini lecture, and we did say that they can serve as intramolecular bonds. Well, here it is. I hope that you're starting to, I hope that you're starting to realize that everything in my course is connected, whether that's by design or accident. It's kind of how it moves. The primary structure of a protein, it's just the amino acid chain, the sequence of which is directed by DNA. This structure is kind of like beads on a string, where the beads are the amino acids, and as you see in this picture here, the beads can be different. But it's not so much the beads on a string, it's the sequence of the beads, the sequence of the amino acids that's important, because it's that sequence that's going to allow folding and hydrogen bonding to create that final functional protein shape. Sometimes this amino acid chain can be called a polypeptide chain. You would probably call it that in a chemistry class. And this is because the type of covalent bonds that bind together the amino acids is a peptide bond. This primary structure, this amino acid sequence, is unable to do anything. It simply does not have the final folded functional shape that has one designated function. So the first level of structure would be a sequence of amino acids or a polypeptide chain. The secondary level of protein structure creates 2 common shapes, one that is called an alpha helix, which is what you see right here. This is an alpha helix. And I'm sorry, I'm drawing with my finger because my computer's not exactly correct today. And this is what is called a beta sheet. It's like I'm signing my name when they give you the Apple Pay thing, and you're like, I can't sign my name. So an alpha helix is a region of a protein it's twisted into a tight spiral. The coils of the helix are held together by hydrogen bonds. And I just want to note that it's not a double helix. It's a single helix, an alpha helix. Other regions of proteins can fold up like a paper fan, and those are called beta pleated sheets. The alpha helices and the beta pleated sheets come together and comprise the secondary structure of a protein structure that results from one event of folding. A given protein might have, I don't know, 3 alpha helices and 4 beta pleated sheets, while another has 8 beta pleated sheets and no alpha helices. The combinations are almost limitless, but most proteins have at least some of each of these structures. Once all the helices and sheets are coiled, folded, and locked into place by those hydrogen bonds, the polypeptide takes on a distinct 3-dimensional shape, and it's very different than that string of amino acids. This is what is called the third or the tertiary level of structure and the overall 3-dimensional shape of a protein. The fourth level of protein structure describes interactions among different polypeptides, right? So not all proteins have a quaternary structure. The big ones usually do. But for many proteins, their quaternary structure is their ability to bind or meet up with another protein. Some complex proteins are composed of more than one polypeptide chain, and hemoglobin would be an example of that. Once all the alpha helices and the beta pleated sheets are coiled, folded, and locked into place, the polypeptide takes on a completely distinct third level of structure called the tertiary level of structure. This level of structure contributes to the overall 3-dimensional shape of a protein, but there is one more level. The fourth level of protein structure, or the quaternary level, describes interactions among tertiary levels. Not all proteins have quaternary structures, but the big ones usually do. Some complex proteins are composed of more than one polypeptide chain that bond together to sort of form a superprotein, and that's what you have right here. This is hemoglobin. It is actually made of 2 different polypeptide chains. Here's one of them, and here's the other one. And it consists of 2 of each, and they're all bonded together. And there's actually an iron atom that's placed in here that's able to be loaded up with oxygen. So we see how the protein structure is made through these levels of folding, but we can also deconstruct a protein. This process of changing the protein shape or unraveling it, unfolding it, is called denaturing. And denaturing usually involves a chemical, like an acid, or heat. I love me some eggs. I do. Scrambled. That's how scrambled. When I cook the eggs, I'm denaturing the storage proteins in the egg. When you are denaturing meat, you can do that with a marinade, especially an acidic one. You can denature meat on the barbecue with heat. We can actually denature proteins, making them safe for our consumption without using heat. Fish exposed to acids are fully cooked, and this is what is called sous vide, making the fish safe to eat. So eggs that are exposed to lemon juice are actually safe to eat as well, because the lemon juice cooks the egg proteins much like the heat from my frying pan.